All right, remember that gene therapy is where we fix genetic diseases by replacing a bad version of a gene with a good version of a gene. Transgenic organisms are where we can take DNA from one organism and put it into another to give them different traits. So we do that with things like GMOs, with crops to make them drought resistant or, create their own fertilizer or pesticide. And we can also do that with medicines like insulin. And then DNA fingerprints we can use to identify people, paternity, and so on. So let's look at the steps of how to make a transgenic bacteria for something like insulin production. So, insulin is a protein. So, what we can do is we can take the insulin gene, so the DNA that codes for that protein, and we can use a restriction enzyme to actually cut that particular gene out. We will then cut a plasmid, which remember that's bacterial DNA. And then, because all of this is DNA, I can actually splice in the human gene into the bacterial plasmid. I can get that bacteria to take the plasmid in. And now that that bacteria has that gene, it can use its ribosomes to create that protein, which would be, in this case, human insulin. And then we can extract that, bottle it up, and send it off to the people that need it. So remember, in a DNA fingerprint, we're going to use gel electrophoresis. So once again, we're going to take some DNA. We're going to use restriction enzymes to chop it up into little fragments. And because everyone's DNA is slightly different, the length of all these fragments is also going to be slightly different. So then I'm going to run them out on a gel. So these represent DNA fragments. The biggest fragments are going to be at the top, and the smallest fragments, they travel the fastest, they're going to be at the bottom of the gel. Now, if I'm trying to identify a person, all I got to do is figure out which one is exactly the same. So in this case, it's not 3, it's not 1. Suspect number 2 matches up exactly. With paternity, remember all the fragments in the child have to come from either the dad or the mom. So the best way to do this is you start with the child and the mom and you eliminate those first. And then the ones that remain have to be in the dad. Otherwise, that's not the dad. And then finally, if we do this kind of backwards, if we're looking at this baby, remember every one of the bands has to be found in one individual of each couple. So couple A, neither individual has that band, so they cannot be the parent of this baby. Whereas couple B, one individual of that couple has every one of the child's bands. So that is their parent.